Okay, here we go. So today's topic is transcription translation. We already talked about this a little bit the other day, those of you guys that were tuning in. So this is the whole point of how your DNA controls what you are and what you look like, and even some of your behaviors and things. Like, I know you've heard people say, well, the DNA is the control center of the cell. It controls what you are. It controls everything, right? This is how it actually does the controlling. Okay. So let me kind of sum it up for you really quick, okay? The DNA is in charge of being the code to make proteins. Okay? Proteins have functions depending on their shapes. And just about everything in your body is a protein. Proteins are enzymes. Proteins are hormones. So yeah, it determines when you go through puberty and all sorts of other stuff. Proteins are the color of your skin, the color of your eyes, the color of your hair. Right? Proteins are your skin. Right? Proteins are your toenails, your eyeballs. Like, all parts of you are made up of these proteins. So the DNA is the code to make specific shaped proteins, which give you the characteristics you have, right? Like, for instance, how do you end up with curly hair versus straight hair? The code on your DNA codes for a protein that, when it's done being built, happens to have a little curl in it, gives you curly hair. Or it's straight, gives you straight hair. Or it's shaped in such a way that when light hits it, the light that bounces back is blonde light, or is black light, or is brown light, and you get different hair colors. So that's how your DNA controls everything. Essentially, what we're going to talk about today is known as the central dogma of biology. And this is all about how DNA carries out the instructions for life. Okay? So, as I mentioned just a moment ago, this is called the central dogma of biology. In other words, dogma is usually a word that we use for belief systems, right? So in saying that this is the dogma of biology, we're saying that this is utterly important. This is our understanding of how everything works. This is the whole process. And it goes like this. And you will be asked about this on the test, I guarantee you. You're welcome to take notes as we go and use that stuff on the test if you like. I'm going to post this video when we're done here. But DNA, we start with DNA. We go through a process called transcription. Okay? It's like a copying process. Then we make mRNA from the DNA. That's the transcribed DNA is messenger RNA. That's what the M stands for. Then we go through a process called translation, where we translate this messenger RNA code into the actual thing that we need, which is a protein. Okay? Right? Please keep those up, guys. So DNA, messenger RNA, protein, transcription, translation. Those are really similar words, so you got to keep them straight in your head. How do you remember it? You might remember that, like in the Middle Ages, monks used to literally transcribe the Bible, right? That was how you got a new copy before there were printing presses. Somebody had to sit down and copy word for word, okay? And that's kind of similar to what's going on here. We're making a copy of the DNA with a few little differences, okay? In translation, that's like taking something to a new language, right? So I'm going to translate this code into something that is completely different. And this is even beyond like a new language here. It's a totally different structure altogether. Okay, so that's the central tenets of biology and how we understand how this stuff works. Now let's dive through it here. So what are some differences? Well, first of all, remember DNA is a double helix. It's two strands. There's two backbones and all these rungs in between. Okay, and those rungs have a bunch of bases on them that are the code. Well, messenger RNA is a little different. Messenger RNA is single-stranded. It's just one strand of code, okay? So keep that in mind as we move along here. And those are the bases. We can draw this any number of ways. That's just little shapes to represent what the code is, okay? So what does the RNA molecule actually look like? You will get asked this on the test, I guarantee you, okay? It is made up of a sugar, a phosphate, and a base. And this is what they look like chemically, if we draw their chemical structure. More than likely, when we draw them for you guys, they're just going to be like a circle, little stop sign looking sort of thing, a square. We're going to use like simple shapes. We're not trying to make you memorize the whole chemical structure of these things. Okay, that's AP biology. So what we're after is understanding how the parts fit together. Okay? The nitrogenous base has a bunch of nitrogens. See all the nitrogens on it? The base is the actual code. This is the thing that is the A, the C, the U, or the G in RNA. Okay? The sugar in RNA is ribose. It's not important that you remember the structure, but it's ribose. So RNA's name is ribonucleic acid. You get it? It has to do with nuclear stuff, like the nucleus, right? It technically is an acid, and its sugar is ribose, ribonucleic acid. DNA, anybody know what DNA stands for? It's really similar. Deoxyribonucleic acid. Ha! See how similar they are? So deoxyribose is the sugar in DNA, whereas ribose is the sugar in RNA. Okay. The phosphate group, okay, this circle here, is actually what is the string. In DNA, it's the backbone. It's the bone that's holding all together, all the bases dangling off of it that are dangling off the sugars. Okay. There's a lot of ways we can draw this stuff. You'll see a lot of simplified representations because we don't want to put out the entire chemical formula every time. Okay, so transcription, the first step, how does this work? The DNA is in the nucleus. Messenger RNA is transcribed, like copied from the DNA. And the messenger RNA is what can get out of the nucleus. It's going to leave the nucleus, okay, and it's going to find this thing called a ribosome to do the next step. Now, how does DNA differ from RNA? Well, here are some big differences here that you will certainly see on the test. In RNA, you never have a T. You never have thymine. Instead, you have U's, which are called uracil. Okay, remember the bases of DNA that we talked about the other day, A's, T's, C's, and G's. Okay, you have all of those in RNA, except you don't have T's. Anywhere there's a T, there's got to be a U. I know it seems strange, like we can't make this stuff up. It's just the way nature actually is. It's how nature is. So it's how we have to just understand how nature operates. If we want to go delve back into why does RNA have U's instead of T's, right? We could actually start to talk about some really complex biology. For instance, how life evolved on this planet in the first place. And there's very good reasoning to suggest that life on our planet was not DNA-based to start with. Life was probably RNA-based. RNA is an inherently more unstable molecule. You know this because you hear about the vaccine, right? Hear about the RNA vaccines that have come out, the only two that have been approved in America so far, right? The Moderna and the Pfizer vaccines. They have to be kept at sub-zero temperatures. Why? Because they're RNA. And RNA is not that stable. It's hard to stabilize. It doesn't last very long. But DNA is a much more stable molecule. DNA more than likely evolved from RNA. And now everything that's alive has DNA. But we also still use RNA. Okay? So what's different here? U's got to replace T's in RNA. A's going to pair with a U. G's going to pair with a C, so that doesn't change. And of course, we have the ribose sugar. Okay? All right. There's another chemical drawing of this. Okay? We've got two bases here. And there's two bonds, little dashed lines between them right there. Okay, so this would be an A and a U combined up here. You don't need to know that. Okay, so what does this look like? What goes on inside your nucleus of your cell is that some of the DNA unwinds. Not all of it. You don't need to use all the DNA. Think about it. If you're a skin cell and you need to make skin proteins, well, you shouldn't be making, something that you need in your kidneys, right? You need to make the parts that are important for the part of the cell that we need to use. So what happens is part of your DNA unwinds. You can see the double helix here, the ladder structure of DNA. DNA is blue up here. And it pops apart, pop, 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 at these weak hydrogen bonds. Only one side of the DNA gets used. It's referred to as the template strand, okay? So along the template strand, the RNA starts to get built. Check it out. G's pair with C's. U's pair with A's. See that? We can't have a T up there. C's pair with G's. If there's a T on the DNA, there's going to be an A on the RNA. Don't goof that up and put a U. That's really common for students to do. You can have an A on the RNA. And you make this long strand, okay, of RNA. The RNA, it's small. It's single-stranded. It can fit out these nuclear pores that are around your nucleus. So it can escape the nucleus and carry the code out, right? Once it gets out, it can then take part in the next step, which is translation. Okay, so this is transcription. We're making this messenger RNA. We're transcribing. See how we're just copying the code with a few differences? Instead of T's, we've got U's on the RNA now, and it's single-stranded. Then the DNA can zip back up and be nice and safe in the nucleus. You want to keep that stuff safe. Why? It's your genetic code, right? You need to keep that stuff safe and stable. The RNA, you can use that, ship it off and use it to do stuff. When it breaks down, who cares? Okay? So translation is the next step. I'm going to keep showing you different representations of this translation step because it's really not that complicated to grasp, but I realize there's a lot of pictures of different stuff up here. Okay? So first of all, what's going on here? Let's look at the picture. In this picture, my messenger RNA that's left the nucleus, it's carrying the message. It's going to find this thing called a ribosome. Now, ribosomes kind of look, remember when we did our unit on cell organelles, and you drew them as little dots? Draw those little dots. If you zoom in on those little dots, they actually kind of look like little Pac-Man looking things. So there's a top part and a bottom part. And we usually draw them kind of as like a little ball. A ball on top and a ball on bottom. You can see the ribosome here in the picture. So what the ribosome does is it chompers onto the RNA strand. And it's going to read it. It's going to slide down the strand and read it. Okay? Now there's some important pieces here. It reads it in 3-letter chunks. It always reads 3 letters at a time. And those 3 letters have a special word that we call them called a codon. So in this picture, A, U, and G are the first codon. Get it? The next codon would be G, C, A, and then U, A, C. You read it in these pieces. You can almost think of it like a sentence in English. It has to be in these chunks, okay? Now, the rest of what's going on in here, it looks pretty complex, but let's break it down. There is this thing called a transfer RNA, tRNA. It's RNA as well, which also means it's single-stranded. So check it out. I'm going to unpack a lot here for you guys. This thing, you see the strand here? It's actually just a piece of RNA. But it's wrapped up and kind of stuck together in a weird way so that the bottom part of it turns out, and we've blown up those bottom 3 bases. And in this first one here, we've got a U, an A, and a C. Well, those happen to pair to that codon down there, the A, U, and G. And we refer to those 3 letters that are on the transfer RNA as an anticodon. Get it? It's anti the codon. It fits, it pairs with it. Okay? Really try to drink all this in, guys, because this unit is a lot more complex than a lot of other stuff we've talked about. Like, you're either going to be able to do it or you're not going to be able to do it. Okay? If you're looking at your phone, playing video games, texting people, listening to music, I hate to tell you, you're not going to be able to do it. Right? That's just the bare facts of this. It's a lot more complex than other stuff we've talked about. It'll either make sense to you or you might as well give up because you're not paying attention. So here we have this RNA, this transfer RNA. Look at the pieces of it here. Okay. There's on the end of it, there's this ball up here called an amino acid. Well, your proteins are actually just chains, big chains of these amino acids stuck together. So the transfer RNA actually carries the amino acids down in to build the chain. So once this transfer RNA fits onto this ribosome, the ribosome holds it in place there. The ribosome is going to snatch that amino acid off of it. Now those are specific. They're specific to the type of transfer RNA, so you could kind of tag them based on their anticodon, right? Or we could tag them based on the codon, because it's always going to fit the same one. So how do I know which amino acid that is? Right? I can absolutely know, because if I know the codon AUG or the anticodon UAC, I know that's methionine, because I can get a table and I can look up what that means. And we're going to do that today. Okay, so be careful. The tables that we give you are always the codon. They're not the anticodon. Sometimes we'll ask you for the anticodon. Sometimes we'll give you the anticodon and ask you for the codon. We'll mess with you. We'll go both directions, even though nature doesn't really do that. But the tables that we give you ask for the codon. They want to know, what does AUG code for? And you'd figure out what type of amino acid would be carried in for that. Okay, so let's go back to our story. Once this guy is stuck on here, okay, the ribosome is going to hold on to that amino acid. It's going to set the transfer RNA free, woo, back into the liquidy cytoplasm of the cell to go pick up a new amino acid. It slides down, the ribosome does, reads GCA. That calls for a different amino acid, a different transfer RNA, comes in, bringing a new one, and it grabs the amino acid and chains it to the last one. See how we're building a chain of amino acids? Okay. A chain of amino acids is going to be let go when we're done with this whole process. It's going to fold up into a special shape, and we're going to call it a protein, an enzyme, a hormone. Okay, and it's going to be you. Sometimes you'll hear us call this chain of amino acids a polypeptide chain, because the bonds that hold the amino acids together are peptide bonds. Get it? So there's a lot of ways to kind of refer to the same stuff here. So don't get confused by it. Just ask if you have questions, okay? So we'll slide down. We'll build this long chain of amino acids. That's your protein or your polypeptide when we're done. How does the ribosome know where to start? It's a good question. Every single one of these strands of messenger RNA has to contain a start codon, a codon that literally says start, and it's AUG. That's one of them, okay? That means start, and so it grabs on at the start, and it puts down the start amino acid. When it gets to the end of the strand, there's going to be another codon that codes stop, and no amino acid gets put down there, but it just lets go. Okay, so that's how it knows where to start and stop. I'm personifying the ribosome here, right? They're not thinking little beings, right? This is what they do, right? So anyway, this is the step. This is what we're doing here. This is what all of today is about. How do you do it? You have to read a codon table. There are two main ways, the two main types of codon tables. They're the same thing. You can use whichever one's easiest for you. Here, I'll show you this one first, and I'll show you the other one. Let's say you have a codon. Let's say I give you the messenger RNA reads ACG. I wrote that up there. And I want to know the amino acid. What amino acid is that going to get put down into the protein chain, to the chain of amino acids? Well, using this table, the first letter over here on the left is A. So I find A here. That means it can be anywhere in this row. It's circled in red. It's squared off in red. Anywhere in there. But the second letter is C. I have to go up to the top of the table where there's a C, and that means it can be anywhere in this column, okay? Do you see how that puts me in this box here? So now I've got choices. It could be ACU, ACC, ACA, or ACG. Could be any of those, okay? I need to look at the third letter over here, and that comes from the right-hand side of this graph, this chart, and it's G. So I find G, and I pull G over, and I have ACG. Okay, that's exactly what I'm looking for, ACG. That's how you read this table rather than trying to look through the entire thing to find ACG, which would take some time. Okay, now what amino acid is it going to give me? That's abbreviated up here, THR. Okay, so usually we only capitalize the first letter so you don't get confused and think that you've written down some sort of a codon or something, okay? But that stands for, it's abbreviation for threonine, which is the type, which is the amino acid. Do you notice here there's a takeaway? There's multiple ways you could get threonine, okay? There's multiple ways above that you could get proline. Serine has multiple ways that you could get it. In fact, multiple boxes on this thing could give you serine. Over here on the right, there's totally different ways you could get the same exact thing put down. So check it out, because it's going to come back later. You could technically change some of your DNA and still end up with exactly the same protein that you need. Like you could have a mutation and nothing could be different. See how that could work out? I mean, it'd have to be perfect, but it could happen, right? And it does happen. Okay, so that's how you read this one. Let's look at the next one. Most folks prefer this. It's like a bullseye. So in this one, you just read from the inside out. If the first letter is A, you start with A, which is right here circled in red. The next letter is C. Well, you find C, which is also within that bigger block of A, which is circled in green. Then you find G, which is the next breakdown, and then that sticks you at threonine. There you go. Okay, and it's little abbreviations up there and whatnot. Okay. So notice also, let's look at some of the other things on this just so you can see them. We've got some stop codons here. Okay, as we look around, down near the bottom there, there's a stop here. Okay, it's not going to put down an amino acid. How do I get a stop? What would be the codon for this stop right here? Could somebody read it to me? What's the first letter? U, and then what? G, A. UGA means stop. Always means stop. But there's another one down here. What's the codon for this one? There's two different ones. UAG or UAA would all give me stop. So there's three different stop codons. Okay. See if that has that labeled up there. There we go. MET up here at the top, methionine, is the start codon, okay, MET, and it's labeled AUG. That's the one we did in the last little thing there. Okay, so see how we can work that out? You're going to have to do that today. Okay, so let's see if you remember the parts here. This is a simplified drawing of the whole process of translation. What is A up here that I'm representing? Anybody? What is A? It's this long strand. Remember what that's called? I'll give you a hint. It's the message. This is the messenger RNA. That's what A is, the messenger RNA. What's B? It's got two parts and it's clamped onto the messenger RNA. Starts with an R. That's the ribosome. This is a picture right off of a test. Okay? Off the top of the ribosome is being built this chain here. Look at this thing right here. What are the individual pieces of that chain called? C is labeling one of them right there. They start with an A. Those are your amino acids. Okay? And then I could call this a number of things. Right now, I'd probably call it a polypeptide chain, okay, or a chain of amino acids. When it's all done, it's going to be a protein. Right? See how that stuff forms, how it kind of moves and puts together? Okay. So let's just kind of refresh it again here. Your DNA is in the nucleus. It undergoes its regular replication to copy, which was mitosis that we learned all about. Okay? But aside from that, it also has to function, not just copy itself. So it does transcription to make RNA. The RNA gets out of the nucleus into the cytoplasm of the cell where translation happens, which is make a protein. How much of your DNA do you think actually does coding? Actually codes for proteins that you need? Think it's a lot, little, about half? Strangely enough, it's only about 1%. You got a lot of DNA in there, and only about 1% of it codes for stuff that you need. Now, it's not that you don't need the rest of it. There are vast regions of DNA that are not coding, that are kind of buffering your DNA and keeping it nice and safe, where mutations do occur and it doesn't screw anything up. There's also regions of your DNA that are called promoters that actually help to initiate different sections of DNA being transcribed and translated. Right? There's also sections that help to do further modifications of things. Like it's a very complex picture in there. So when you do see that only about 1% of your DNA is like important for doing stuff, it's not really the whole picture. But yeah, like most of your DNA is not actually for coding just proteins. So it's what we're discovering is that there's way more complexity going on in there than anybody first thought. Okay, so what else occurs? Well, mutations also happen to your DNA. This is the primary way that we get new characteristics in terms of evolutionary change. How does the coronavirus keep changing, right? Well, mutations. Mutations to its genetic code cause it to change. And we're going to look at some of the different ways that can happen here. Well, errors can happen during replication when you're copying your DNA, mitosis, okay? Or even meiosis when you're making sex cells. And that's how they'd get passed down to the children if we're talking about humans. They can also happen during transcription and translation. Your proteins can get screwed up there, okay? Another way mutations can happen is from mutagens. Let's say if you're exposed to some radioactive material, or let's just say you're exposed to some sunlight, like too much sunlight, or you like to sit in a tanning bed, right? That can mutate your DNA. Ultraviolet radiation from the sun can change your code. And sometimes it can be really bad, like skin cancer, right? So these mistakes happen. There's a lot of different damage that can occur, but sometimes they're useful. Right? Oftentimes, I'd say the majority of times, nothing happens at all. And your cells have machinery for repairing this stuff. So it's not end of the world if a single cell has a mutation. Like, your body has ways of dealing with this. Okay? So let's look at the two main types of mutations, because you'll need to know those for the test and for our activity. Okay? The first one's called a point mutation. And that's where just one base changes. An A or a T or a C or a G changes. Okay? So let's take this sentence here. Let's say I had a point mutation in the sentence, the dog bit the cat. See how I've broken it up into 3-letter chunks? Okay, so it's kind of like codons. This is not really codons. A mutation that would be a point mutation would look like this. You see the difference? How many letters did we change? Only one. But it says something really different, doesn't it? The dog bit the car is different than the dog bit the cat. So maybe we just created a non-functioning protein. Maybe we created a better functioning protein. Maybe we created a worse functioning protein, but it's still good enough that you're not going to die. Okay? So one base pair change is only going to change the meaning of a single codon, which means it's not going to change the shape of the resulting protein a whole lot, is it? It might change it a little bit. Now, it could be disastrous. It could be helpful, but it might not be a big deal. Okay? It's a point mutation. By the way, skin cancer is usually caused by a point mutation, one letter flipping from exposure to ultraviolet radiation. So it can be real bad. Okay? Let's look at how it plays out on an actual codon. So here's a strand of messenger RNA. The resulting protein is down below that we're writing out. So each 3 letters of that codes for something. We use the table to look that up. Okay? This is the original normal one. There's, of course, a stop there at the end. If we look at the next one, the G here got flipped with an A. It got replaced. That's a point mutation. Now, instead of glycine, we're putting down serine. Is it going to be shaped right, or do we just change your hair color? Right? Like, where it happens and what it does, it could be different now. Okay, that's a point mutation. Let's look at the other type. The other type of mutation can, and typically is, more disastrous. It's known as a frame shift. We totally change the reading frame of the sentence. So there's 2 ways this can happen, and you're going to see both on one of our assignments. You can either add a base that was not there before, or you can delete one out. That's different than just flipping one. Okay, so let's say we've got a sentence again. The dog bit the cat. In a frame shift mutation, this might now read, head ogd id heck at. You've completely jumbled the whole thing. Can you pick out what has been deleted there? Do you see how we lost the T over here on the very beginning of it? If you knock out one letter, everything slides down. So the end of it might not even be a stop codon anymore. In fact, the start of it might not even be a start codon. You could have really had a big screw up here. It could be real bad. It shifts the entire reading frame. So here's one that shows a deletion. So we start with the code up here. Okay. Now notice how much gets screwed up. This is a test question. Is it just one amino acid that gets screwed up? No. Phenylalanine doesn't become just leucine, but everything after phenylalanine is totally jumbled. It's completely different because everything slid down on the reading frame. So a frameshift mutation is definitely going to be a bad deal. Okay, a point mutation, it maybe, maybe not. Okay, but the frameshifts are change a lot of the code. Okay. Okay, so I think that is the end there. Okay, what I've tried to draw for you guys here is a picture that just kind of represents everything that I just talked about. So this is protein synthesis. Okay, this is the central dogma of biology right here. And if you picture that this thing that I drew here, essentially this is a nucleus of a cell, okay? Inside the nucleus is the what? The DNA, right? It's in here. That means that my cell is like bigger than my entire paper. It's like the whole wall that I'm projecting on. It's humongous, okay? So outside of the cell out here is the cytoplasm, the liquidy interior of the cell. So what's going on inside of the cell during transcription translation? Transcription is the first step. There's your DNA right there, okay? In DNA, G's pair with C's. So this was originally paired together. See how it's double helix? I've popped part of it apart. All the hydrogen bonds have broken across here. A's are paired with T's, T's are paired with A's, T's with A's, C's with G's, D's with C's, right? So on one side of this doesn't do anything, but the other side of it, we're going to build messenger RNA. Messenger RNA is a single strand, not double like DNA. Okay, so only looking at the messenger RNA over here, G's are going to make paired up with C, A's will pair with T, C's will pair with G, G with C, A with T, A with T. But down here, if I have an A on the DNA, I can't have T's, right? So I'm going to have a U, uracil. So now once this is built, this can break off. This was transcription step, DNA to RNA. Now it can break off, pop, 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 and it's little, and it can leave, boop, 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 out the nuclear pore in the nucleus. So this is a membrane that's holding all the DNA in, okay? That's what the nucleus is. So the DNA then can wrap back up and get nice and safe and stable, and we keep our code safe. So the part that we need just comes out, a little copy of it called transcription. It's going to go over here, and it's going to locate this thing called a ribosome, okay? The ribosomes are out there in the cytoplasm, the liquid of the cell. They could be on the refa endoplasmic reticulum. These guys are going to grab a hold of this strand of messenger RNA. They kind of look like a Pac-Man. There's a top part, and there's a bottom part. All right. By the way, a ribosome is actually made out of RNA. If you're wondering what it is, it's actually these strands of RNA all wrapped up. Okay, it's called rRNA. Get it? Ribosomal RNA. So RNA has a lot of uses in the cell. Okay, ribosome, chompers on. The ribosome is going to read every 3 letters, because 3 letters are a codon. The codon here is GAA. I could use my table to figure out the amino acid. Remember the codon table? I'd need GAA and I could sort it out. But what's really going on here in this step of translation? In translation, we're taking the messenger RNA code, we're translating it into a protein, which is a chain of amino acids. So where do they come from? These transfer RNAs are out here in the cytoplasm, moving around, okay? Each transfer RNA has a specific anti-codon that will match up with a specific codon down here on the code somewhere. There's a lot of these things. Each one carries an amino acid, a specific one. Methionine, serine, whatever you have, okay? They're specific to their anti-codon, which is specific to the codon. So this thing comes in here, plunk, connects to the code that it fits with. G with C, A with U, A with U. Remember, there still can't be T's. We're dealing with transfer RNA. On the end of this thing's an amino acid. I didn't write down which kind. It peptide bonds to the one that was there previously. Now the transfer RNA goes free. Ribosome slides on down. The new one comes in, binds on. We stick it together, and this chain of amino acids just keeps growing out of the top of the ribosome. Okay? When it's all done, it will hit stop, it will let go, and this will wrap up into our protein. Central dogma of DNA, of biology. DNA transcribed to RNA, messenger RNA. The message leaves the nucleus, finds a ribosome, where the messenger RNA is then translated into a chain of peptides, a peptide chain, an amino acid chain, amino acid sequence, which is the protein. Okay? Transcription, translation. Boom, done. Looks like the whole unit. Okay, take a close look at it. It's got everything in it.